All right. Hey guys. Welcome to my first ever pre recorded video lecture. Oh my God. Um, Hopefully this gets a little bit less uncomfortable for me. Okay, so we're going to do Mammal Transport Part 2 today. So it's about arteries and veins and their structure. And then it gets a little bit into like hemoglobin and how hemoglobin actually carries oxygen. So let's do it. Okay, so we've already talked about arteries and veins. So arteries carry blood away from the heart. I do have my little stylus. Arteries carry blood away from the heart, which means they're usually going to be oxygenated. Okay, can't really write. Or red. Except that pulmonary artery, because remember that's going to the lungs to get oxygenated. Veins are coming back to the heart. So they're usually deoxygenated, usually blue. Then we've got capillaries in the middle here. I think we've already even labeled that those are for gas exchange. All right, not quite gas exchange. Not quite as responsive as the smartboard here. So we kind of go from arteries, those branch down into capillaries. Capillaries are going to empty into veins, and we're going to head back to the heart, and we've got that whole, closed double circulation going on. So arteries and veins have similarities and differences in their structure. They each have 3 layers. I'm going to put a star here. So the endothelium, endo means inside. So endothelium is going to be the innermost layer. Tunica media. Tunica means lining. So the tunica media is kind of the lining layer in the middle. It can have things like smooth muscle to contract or relax. It also has a lot of connective tissue. Things like collagen, really stretchy because of the pressure of the blood, and these vessels need to be able to stretch. And then tunica externa. Externa is going to be the outer layer. That's mostly just stretchy connective tissue to kind of hold everything else in. Capillaries, since that is where gas exchange again is actually happening, you want to minimize the distance that things like oxygen and carbon dioxide have to cross. So they only have 1 layer of endothelium, no middle or externa or anything like that. So here we have a diagram. So endothelium in the middle, the tunica media. Notice it is a lot thicker in arteries than it is in veins. That's going to come back to pressure. When you think about the pressure that the blood is under as it's being pumped out of the heart, arteries are going to have to handle a lot more pressure than veins because, the blood is coming from the heart. So thicker tunica media, the externa is about the same. Also notice there's some valves in those veins because when you have less pressure, backflow becomes a problem that you have to deal with. Okie dokie. So arteries are moving blood at high pressure away from the heart, so they have a really thick tunica media to withstand pressure. In that picture, it's that whole little piece there. So these really thick walls, lots of pressure. It also has smooth muscle there that can contract and relax to help regulate blood flow. When you're when you're embarrassed and your face turns red, that's partially those blood vessels dilating to increase blood flow. I don't know where I was going with that. I wish I could go back and erase things, but I can't. Or if you had a trauma, if you cut yourself really badly, the vessels leading to that area can contract to prevent blood loss. So being able to some extent regulate that blood flow is really, really important, and that's why we've got muscle there. So big arteries are going to narrow into arterioles, and then that's going to go into the capillaries. So notice that thicker wall. Oh, here's another picture. Kind of breaks it down a little bit more. Okay, so veins are moving blood at a lower pressure back to the heart. They've got thinner walls, so this looks a little misleading, but the wall itself isn't as thick. It doesn't have as much muscle. They do have semilunar valves. Remember, those have that trash can whoosh shape. And normally they're not going to be as perfectly circular either. When you think about a hose that has water flowing through it, it's going to be a lot squishier if there's not a lot of water flowing through it. So more pressure in the arteries means they're going to end up being more circular. Veins tend to have more of this like floppy, not pressurized hose look to them. Another picture. Okay, so here's a comparison chart. So arteries are under a high pressure. They have a smaller diameter and a narrow lumen. Those both, the stylus isn't great, maintain that pressure. Imagine trying to bring water up through like a straw versus a hose. The smaller lumen or the smaller space in the middle is going to allow that pressure to stay higher for longer, the blood to get further. Really thick, muscular tunica media, elastic tunica externa. So when you actually like feel your pulse in your neck, that's the wall of that artery stretching. So really strong, really stretchy walls. And no valves because the pressure is so high that you're not worried about backflow. Veins, we've got a lower pressure with a larger diameter. Again, not as much pressure. They don't need to maintain that pressure. So everything else is a little bit thinner, a little bit less elastic. Semilunar valves prevent backflow. We're always trying to keep the pressure up and prevent backflow. All right, here's a picture. So we've got an artery here. I wonder if it'd be better if I just use my mouse. And then this would be a vein. Nope, not really better. So artery here, you can see the really thick wall, circular, smaller lumen. This one also really compares like the wall artery versus vein a little bit better. So thinner walls, a lot bigger lumen, also a lot less circular. So plan diagrams. This is a big emphasis with plants. So hopefully we're back in the classroom by the time we do that. But technically speaking, that objective says observe and make plan diagrams of the structure of arteries, veins, and capillaries using prepared slides. So technically speaking, this could be one of the slides that you get on your ACE exam. Realistically, they've never done it because plant slides are a lot cheaper than arteries and veins. So technically it could happen, probably not. But plan diagrams, we talked about them a little bit at the beginning of the year. It's just using lines to show tissue distribution. So in an artery, you'd see thicker walls, smaller lumen. Vein has a bigger lumen, thinner walls. Hopefully, if we're back in the classroom, we can look at these under the microscope. But for now, just keep that in mind. So capillaries are between the two. Their job is to carry blood as close as possible to every single cell in your body. They're extremely thin, about 7 nanometers in diameter, which is only one red blood cell. So at some point, you have red blood cells flowing single file through your capillaries. Wow. And they just have one single layer of endothelium because, again, you want to minimize the distance for diffusion. It's a pretty nice diagram. Oh, you can color code this if you printed out your notes. So from the heart, we have arteries. Arteries branch into arterioles, branch into capillaries. And this is where gas exchange happens. I'll change the color of my pen. Capillaries go to venules, which are just little veins, into the big veins, and then back to the heart. Okay, the lymphatic system. So lymphatic should sound like lymph to you, should sound like lymphocytes. It's pretty much another set of vessels for lymphocytes and antibodies and things like that to get around the body. It's like an express lane for these cells, essentially. So lymph, as far as like what it's made of, it's a fluid similar to blood plasma. So blood plasma is the liquid component of your blood. So mostly water, probably like 90-something% water. A little bit of protein, a little bit of salt, a little bit of sugar. Mostly water. Transport proteins like antibodies. It can also grab bacteria and bring them to your lymph nodes. That's what's like right under your ears when you're sick and you're like, oh, can you feel your lymph nodes? Or why the doctor would like feel that top part of your neck. Because if your lymph nodes are swollen, then you're probably dealing with an infection. So just essentially, an extra highway. It does have places where it attaches to your main bloodstream to get things from point A to point B, but it's an extra set of vessels for the transport of antibodies, essentially. Okay, so state and explain differences between blood, tissue, fluid, and lymph. Blood is your blood. So blood vessels in the heart. It's plasma and cells. Sometimes they call these formed elements. Formed elements or cells. Tissue fluid is the free-floating fluid in your body. It's got, again, a lot of similarities with blood plasma, but it doesn't have the cells or certain proteins because most cells and proteins are too big. So really just the juice from your blood. And that's just because cellular processes, things like diffusion, gas exchange, tends to happen better in moist environments. We like to be squishy. Okay, so tissue fluid happens when you have these capillaries, and because of the pressure pushing against the walls of those capillaries, some of the water in the fluid will push out through that wall into the tissue. So it can go out, it can also come back in this way. So pretty much anything that's in the blood or in the plasma of the blood rather, that's small enough to pass through, can be part of tissue fluid, is going to get pushed in and out. But again, most cells, proteins are too big to pass through the capillaries. So tissue fluid doesn't typically have cells, doesn't typically have some of the bigger proteins. Okay, blood composition. So again, here's the plasma. All this stuff in it, pretty much just water with other stuff in it. Red blood cells, I don't think you need to know these numbers, but about 45% red blood cells. I think like 55% is plasma, and then it's going to be less than 1% white blood cells and platelets. So really, mostly red blood cells, mostly plasma. Functions of blood seem pretty straightforward. Transport things, regulate pH, body temperature, especially in things like mammals, we have a pretty set body temperature. The blood flow helps to do that. Protection, your immune system, clot formation can also help with wounds and infections, stuff like that. Ooh, 3 important proteins that live in your plasma. Well, they don't live, but 3 important proteins in your blood plasma. Albumin regulates water movement between tissues and blood. You don't want your blood to be too watery, too diluted, or too concentrated, not watery enough. So albumin can actually kind of absorb and let go of water as needed. This is also what makes egg whites have their consistency. Globulins are immune system and transport. Antibodies are called immunoglobulins or immunoglobulins. And then fibrinogen helps with blood clot formation. That can be good or bad, depending. But when you get like a scab or something like that, fibrinogen is helping out with those platelets. Okay, so some blood cells. So red blood cells are biconcave. Typo. Yeah, biconcave. And anucleate. That means that they do not have a nucleus. Are you serious? Okay, so that means that they do not have a nucleus. When they're dividing from the stem cells in your bone marrow, obviously they have to start with a nucleus, but it breaks down. Main components, hemoglobin, British spelling, lipids, ATP, carbonic, anhydrase. ASE must be an enzyme. And they transport oxygen and CO2. One thing I do want to point out here is that 7 micrometers in diameter. If you get asked a short answer question about red blood cells or describing red blood cells, that 7 micrometer diameter can get you a point most of the time. So red blood cells, they are biconcave, which means that they kind of loop in at the center. So if you're looking at it in a picture, the center does tend to be a little bit lighter, like right in here. And if you're looking at like a blood smear, microscope slide, which again, hopefully we can do. But if you're looking at it, it's going to be mostly red blood cells. Here we see some white blood cells, but everything else is red. Ooh. So technically, this objective, monocytes, neutrophils, lymphocytes, is an objective from this chapter, but I threw it in with the immune system because we were actually talking about what those cells do. But don't forget them. So monocytes have the kidney bean. This is a lymphocyte, small, dark, dense nucleus. And then remember, neutrophils have that lobed nucleus. Okay, so now hemoglobin. Big part of this chapter. So hemoglobin, we've got 4 hemes and 4 globins. The globin molecules transport CO2. The heme molecules transport oxygen with the help of that iron ion. So if you're iron deficient, your body may not be able to carry oxygen very well. It is a globular protein. Remember, all of our proteins are either fibrous, long, skinny, into fibers, or they're globs. This is a glob. They have done in the past locate with arrows the location of the iron. So it's all of these groups here. It'll normally look like a circle. So each heme group, and you have 4 of them, each heme group can combine with 1 oxygen. So a saturated hemoglobin can carry 4 oxygen molecules. A saturated hemoglobin molecule is called oxyhemoglobin. All these big, long hemoglobin words are going to become really important, so keep an eye on it. Okay, so this is the oxygen dissociation curve. Isn't it beautiful? So this curve, and I'm going to give this 3 stars. Holy cannoli. So it shows you the percent saturation of hemoglobin, so how much of your hemoglobin is saturated at different partial pressures of oxygen. Partial pressure of oxygen is really just how much oxygen you have. So you see it kind of starts off slow and then it gets really steep there before kind of leveling out. And that's because hemoglobin has this really cool trick where it changes its conformation to make each additional hemoglobin bind easier. So if you have regular hemoglobin, first oxygen binds, it's going to change shape just a little bit to make it easier for oxygen number 2. It changes shape again. Oxygen number 3 is even easier than 2, and it keeps going until you're saturated. So once you get that momentum going, once you have that like half saturated hemoglobin, it's going to be easier to saturate it. And again, that is called oxyhemoglobin. So what this curve tells you, and it does kind of make sense, is that when there's low oxygen, which I can keep the highlighter. So at low oxygen, say like 2 kPa, you've got maybe 20% of your hemoglobin saturated. So there's lots of uncombined hemoglobin, not very much oxyhemoglobin. In areas where there's high CO2, or high oxygen, say 14, wow, you've got just about all of your hemoglobin saturated. So when more oxygen is available, the hemoglobin will be more saturated. That makes sense. The Bohr shift. So this guy, this is Christian Bohr. Fun fact, he's not the same guy that did the Bohr model that we talked about in chemistry. He's that guy's dad. Talk about a family legacy, am I right? Okay, so the Bohr shift is pretty much saying that, okay, this is oxygen, and this is the oxygen curve, and this is how it works. But that's not the only thing that you have to look at when you're talking about, gas exchange in your tissues. CO2 is also going to play a role. So carbon dioxide plays a role here, and this Bohr shift, it's going to shift the curve, is what he was talking about. So while some of the carbon dioxide does just hang out in your blood plasma, some of it diffuses into the red blood cells, where it's going to affect the hemoglobin. It's going to affect how the oxygen binds. Whew, this is a really, I'm going to add some stars here too. Just star happy today. So remember, carbonic anhydrase is an enzyme. It's in the cytoplasm of red blood cells that catalyzes this reaction. So when you do have carbon dioxide entering your red blood cells, this enzyme is going to catalyze the conversion of CO2 into carbonic acid. And then carbonic acid breaks down into bicarbonate HCO3- plus hydrogen ions. We're going to look at what those hydrogen ions do first. And I want to point out that these double-ended arrows here and here, just means that reaction can go both ways. It's kind of flipping back and forth continuously. We're not super worried about that. So we're going to look at those hydrogen ions first. So this happens. This enzyme is here for a reason. What we're trying to figure out is why. So first with those hydrogen ions, hemoglobin will super readily grab onto those hydrogen ions. When you're increasing the hydrogen ions, you're making an acid. So that is hemoglobinic acid. I swear they will ask you a question where like blah, blah, blah something. One answer choice is oxyhemoglobin, one is hemoglobinic acid, one is going to be carboxyhemoglobin. Then we're also going to talk about carbaminohemoglobin. So you got to know those big hemoglobin words. So hemoglobin will readily combine with the hydrogen ions, gives you hemoglobinic acid. Hemoglobinic acid has a low affinity for oxygen. Affinity is a measurement or like a measure of how likely you are to grab onto and hold onto something. I have a high affinity for double stuffed Oreos, less of an affinity for Brussels sprouts. So hemoglobinic acid has a low affinity for oxygen. So if it happens to have any oxygen, it's probably going to let go of it once it becomes hemoglobinic acid. This also helps regulate blood pH. Excess hydrogen ions in your blood is going to increase the acidity of your blood. By having this reaction happen and hemoglobin's ability to grab onto that, it's going to kind of stop the free-flowing hydrogen ions from affecting your overall blood pH. Okay, so the Bohr shift is saying that when there's more carbon dioxide in the blood, you have more hemoglobinic acid forming. That makes sense. More hemoglobinic acid forming, we're releasing more oxygen into our tissues. Also makes sense. In tissues that are generating a lot of CO2, that means they're doing a lot of self-respiration. That means they need a lot of oxygen. So it's actually a really, really, really like intuitive feedback loop when you think about it. So if we're applying this to our curve, it's going to shift the curve down and to the right because hemoglobin releases oxygen more readily. So that's what this curve looks like. So say at 40, where you've got like 22, whatever, 22% saturation at 20 partial pressure of oxygen. When you up that CO2 concentration to 80, now you've got maybe 16% saturation. You're letting go of the oxygen. It's releasing the oxygen in the tissues that are going to need it the most. So back to this beautiful equation. What's going to happen to those bicarbonate ions? About 85% of the carbon dioxide in your blood is transported as bicarbonate ions. It just gets released back and it goes to the lungs and gets exhaled and all that. So 5% of the rest of that just stays in the blood plasma as carbon dioxide, nothing happens to it. The other 10% diffuses into the red blood cells, is not broken down by the enzyme, and then combines with hemoglobin. Carbaminohemoglobin. Wow. Okay, and the very, very last thing, describe and explain the significance of the higher red blood cell count of humans at higher altitudes. So at higher altitudes, there's less oxygen available in the air. Think like altitude sickness. The higher up you are, the less oxygen's available. So less oxygen's available, that means you have to be more efficient. So mammals at higher altitudes produce a larger number of red blood cells to compensate. I say mammals, sometimes they talk about humans. Cambridge actually likes to talk about llamas. So if you read something about llamas in the mountains, they have more red blood cells. Very last thing I want to do is switch back is switch back super quick to those big hemoglobin words, because there's something in the notes about that. I'm so sorry. One sec. I want 3 and 2. And I want the font to be really big. 3, 5. Okay, so we've got carbonic acid, oxyhemoglobin, hey honey, and carbaminohemoglobin. My husband just walked down the stairs. He's wondering when he's going to get his mic back to play video games. Okay, so carbonic acid is CO2 plus water catalyzed by carbonic anhydrase. Oxyhemoglobin is just hemoglobin plus 4 oxygen molecules. It's a happy hemoglobin. Carbaminohemoglobin is hemoglobin plus plain, not broken down CO2. And I actually want to add one more. I know this isn't super scientific, but I forgot about hemoglobinic acid, which is hemoglobin plus those hydrogen ions left over from that big reaction. So hopefully this went okay. Let me know if you have any questions. And I am so looking forward to seeing you guys soon.